What is gene therapy and how does it work? Illustrations by Mark Hector. As you have probably heard, gene therapy is currently under development for several different metabolic diseases. Some forms of gene therapy have already been approved. However, there is often a lot of controversy about the safety of this treatment and its price, and that is why not all therapies are available in the Netherlands. So what is gene therapy? Is it a drug or a medical treatment? And what can we realistically expect from it in the future for the treatment of metabolic diseases? For some people, gene therapy has a negative connotation. On the other hand, some people expect that gene therapy will cure their condition completely. Within this webinar, we hope to provide you with some basic information. Even if there are no developments for your diseases yet, we hope this presentation will give you a better understanding of how gene therapy works and why it is easier to develop for some conditions than others. So what are we going to talk about today? In the next 20 minutes, we'll start by looking at the developments that led to gene therapy. Then we'll take a deep dive into the cell to see how DNA is used to make proteins and enzymes. After that, we'll look at the methods used to alter DNA and the dilemmas surrounding gene therapy. Hopefully, after that, you should know the basics of gene therapy and can continue to find out more at your own pace. VKS will be happy to provide you with a list of consulted sources and information on specific diseases. Please note that this is a general explanation, and we shall not discuss gene therapy for specific metabolic diseases. How do we actually know how it works, and what has brought us to the point where we can make changes in genes? It all started with the discovery of DNA. A German called Misha discovered nuclein, later called DNA, by dissecting cells in used bandages that were placed on festering wounds. What DNA looked like and its precise construction was not discovered until more than 80 years later, in 1953. While little was actually known about cells, their function and the immune system, the first stem cell transplant took place in 1957. The aim was to give healthy white blood cells to patients with leukemia. New blood cells are made from stem cells in the bone marrow and can be used for transplantation. But it wasn't until 1969 that patients regularly survived a bone marrow transplant. In the beginning, the immune system often ran out of control or the patient suffered severely from the radiation needed to make room for the new bone marrow cells. The first attempt to use gene therapy to treat a metabolic disease actually happened in 1990, when an attempt was made to give healthy cells to treat the illness. Unfortunately, the patient did not survive. The first successful gene therapy product, Glybera, was not available for another 20 years. It was safe enough to enter the market, but was withdrawn again due to a lack of patience. But now, 10 years later, a flood of products is on the way to be marketed as gene therapeutic drugs. To achieve this, a lot of inventions had to be made and brought from theory to practice. Knowledge about DNA and genes, the conversion of genes into proteins, and the function of those proteins in our body has increased very rapidly over the past 4 decades. However, it remains very difficult to use all the available knowledge to help patients suffer less from their disease. There is much more theoretical knowledge than can be tested in human practice. History also shows that practice is often uncompromising. You know what you've got when you have a disease, but you don't know what you get when you intervene in the disease process, at least not until it has been tried in a whole group of patients. On the right of this illustration, you can see a representation of the cell containing DNA. DNA is rolled up very tightly into chromosomes. If you unwind the strands of the chromosome, you find that they contain a double helix, which is the DNA. A specific portion of DNA that contains the code for a protein or function is called a gene. Genes are too small to be seen under a microscope. On the left-hand side of the illustration, you see a diagram of a small piece of DNA. Notice that it is built up of 4 chemicals. These are called nucleotides. Nucleotide A always pairs with T, and C always pairs with G. In the examples below, you can see that a nucleotide on the wrong spot messes up the DNA code. There are different ways in which DNA can become faulty. Whether it is a wrong nucleotide that cannot pair with its counterpart, an insertion of a nucleotide that does not belong, or a deletion of one or a couple of nucleotides. It all results in a gene that has an altered code. Following the bone marrow transplants for leukemia, knowledge progressed rapidly as it was discovered that stem cells can grow into all kinds of cells. When the stem cells of a person with a genetic mutation are replaced by donor stem cells, new cells are produced that do not contain the genetic flaw. Drugs were developed to increase the number of stem cells moving from the bone marrow into the blood. This made it easier to harvest stem cells for stem cell transplants. When the transplant of stem cells is successful, certain enzymes or proteins can be formed again because the donor cells multiply themselves and contain the right gene. You could say that stem cell transplantation is a kind of precursor to gene therapy. Not so long ago, when a good way to suppress the immune response of injected donor cells and organs was not yet known, many patients developed severe rejection symptoms, or they got cancer from the chemotherapy and all the drugs that they had to take. In recent years, treatment by both stem cell transplantation and donor organ transplantation has become much safer and more effective. This is due to all the knowledge that resulted in better medicines and better treatments. For some metabolic diseases, stem cell transplantation or organ transplantation, for example of a liver, is now the standard of care. Now we shall explain how a gene becomes a protein. To be able to explain how things work, I first want to give an impression of the scale on which things take place. Everything is tiny. Cells are not visible to the naked eye, yet together these cells perform all functions and tasks in your body. They do this by enabling all kinds of biochemical reactions. There are many organs and tissues in the body, and they are all made up of cells, 60 trillion in total, together working in an organized manner to support the functioning of your body. Here you see a lot of numbers that give information about the vast amounts of cells and the small scale of everything. The cell consists of a nucleus surrounded by a fluid called cytoplasm. The cytoplasm contains several small particles, for instance the ribosomes, lysosomes and mitochondria. The cell is enclosed by a membrane, allowing substances to move in and out of the cell. The nucleus of the cell contains your DNA, neatly coiled as chromosomes. The nucleus is working constantly. It produces new cells by multiplying the DNA, but it also makes products for the cell. DNA consists of different pieces, the genes. Each gene contains an instruction for making one or more proteins, and each one of us has approximately 20,000 different genes. Genes consist of 4 different molecules, referred to by the letters A, T, C, and G, the nucleotides. So genes consist of nucleotides. The nucleotides are 4 different substances that form base pairs. A always pairs with T, C always pairs with G. Within the nucleus, the DNA is unfolded. RNA polymerase reads half of the strand. This RNA copier completes the base pairs together by placing the opposite nucleotides in a row. This new strand is called RNA. The formed RNA strand leaves the cell nucleus. Ribosomes in the cell use the RNA as a template to form proteins. Ribosomes are located in the cytoplasm. They work as small protein factories by continuously joining sets of 3 nucleotides which form one amino acid. Amino acids are the building blocks of proteins. Long amino acid chains form a specific protein that has a function in the cell. Each combination of building blocks forms a different protein. That protein or enzyme is the executor of a particular biochemical reaction. If the gene has a mutation, there is a change in the letter code. As a result, the nucleotides no longer belong together. The letter order on the RNA also changes so that no functioning protein is formed. This is what often happens in a metabolic disease. A certain gene has a mutation, and as a result, a certain protein or enzyme cannot be formed properly. This has significant consequences for the functioning of cells and therefore for the entire body. If it was possible to get a piece of genetic code without the mutation into the cell nucleus, correct RNA and a correct amino acid could still end up in the right place. Then the protein or enzyme that wasn't working would work again. So how do you get functioning DNA into the nucleus? If there is a disease-causing mutation in a gene in the cell nucleus, we look for a way to change this mutation. We do this by introducing DNA without that mutation into the cell nucleus. Most gene therapies that are currently being developed add a new piece of DNA to your cells. But ways of changing the RNA in the cytoplasm so that a functional protein is built again are also being considered. In order to get replacement DNA into the cell nucleus, you need a transporter that is able to penetrate that nucleus. Remember, the scale at which everything takes place is tiny. We know that some molecules are capable of entering the cell, viruses for example. Unintentionally, we have learned a lot about this in the last few years because we were dealing with the COVID-19 pandemic. A virus is nothing more than a small package of DNA that needs a host, such as a human body, to reproduce. That virus is packed in a shell, a vector, which makes sure that the package can enter the cell unnoticed. For example, if we become ill with a flu virus, the vector carries the virus to the mucosal cells of our respiratory tract. There, the virus injects its DNA into the cells, after which our cells start reading that DNA from the virus, forming RNA that is transported out of the nucleus. Proteins are then made from which new viruses are built, which in turn infect other cells. Only after infection has taken place does our immune system get into action to clear up the virus. That's why you can get a fever and you feel unwell. Vectors that normally transport viruses could thus also bring other DNA into a cell. That is why they are interesting for gene therapy. All parts of the virus are removed from the vector and replaced by a piece of DNA that does not contain a mutation. Thus, the vector no longer contains any viral components to which our immune system would respond and is therefore safe. There are different vector types. Some can transport relatively large pieces of DNA, while others work better in certain cell types. So how exactly are vectors used in gene therapy? Two methods of gene therapy are now in use using vectors. One method is called in vivo gene therapy. The other method is called ex vivo. The difference between the two methods lies in the cell type in which the DNA is modified. Different cell types have different vectors for easy entry into the cells. Here we shall describe the in vivo method. In vivo means in the living or in the living organism. In other words, direct administration to the cells. We shall start at the top left of this illustration. A vector is selected that can enter the cells. Then a good copy of a gene, shown here in green, that you want to replace in the body is introduced into the vector. The patient's immune system must be suppressed to ensure that it will not fight the inserted DNA. These vectors are injected or infused, usually close to the place where they must do their work. There are specific vectors which are known to have a preference for certain cell types. At the bottom right, you can see that the vector wants to enter the cell to bring the genetic code into the cell nucleus. This is preceded by some difficult biochemical tricks of the vector which enable the introduction into the cell nucleus. When the inserted gene is read, the correct protein is made. In ex vivo gene therapy, a patient's stem cells first need to mature, then they are harvested. The stem cells are filtered from the patient's blood. Ex vivo means out of the living or out of the living organism. The stem cells are processed outside the body. As with in vivo gene therapy, a virus is converted into a vector, as can be seen in the top right of this picture. Then the vector is added to the stem cells. Once the stem cells have taken over the inserted piece of DNA, the stem cells are infused back into the patient's body. Stem cells are like carrier pigeons. After entry, they all migrate to the bone marrow to start making new cells. In advance, space is made in the patient's bone marrow by eliminating the already present stem cells with chemotherapy. When the stem cells with the new DNA begin to divide to form other cell types, they all contain the new piece of gene, and so the cells can build proteins again. The advantage of this method is that there is a smaller risk of rejection because the patient's own stem cells are being used. In addition to these two traditional gene therapy methods, much more intricate work is being done on more subtle changes in DNA or RNA using even newer techniques. CRISPR-Cas9, for instance, works like a pair of molecular scissors. It can cut DNA at specific locations and either delete a section or replace it with a different sequence. RNA can also be repaired with the use of zinc finger proteins. These two methods are probably more likely to fall under cell therapy than under gene therapy. Inventing a gene therapy is no longer difficult. We know what we want. We want to put new DNA with the right code in the right place in the nucleus of the cell. We want to make sure that the code is read and that the right protein is made. But how can you do that for so many different diseases? How do you prove it works in a patient? And is it ethically responsible to make changes in human genes? Is it even safe? And what would be a fair price for such a treatment? The entire developmental process of disease-specific gene therapy still takes a very long time. If gene therapy is marketed as a medicine, its effectiveness and safety must already have been demonstrated during research. But we do not yet know how long the effect will last, because patients are only followed for a limited time in the research period, and animal models may not be directly analogous to human response. Once the therapy is on the market, the manufacturer is obligated to continue monitoring the results in order to investigate its effectiveness, safety, and long-term side effects. How much will they charge for this new treatment to recover the costs? Are the health insurers going to compensate these costs? Is the company going to sell it, or are they only concerned with the value of their shares? Some of these questions also arise when a gene therapy is developed by scientists as a procedure to be performed in a hospital. The most important of them is, can the procedure also be performed in other places? Will the quality be the same, and how do you measure that? Will hospital-developed gene therapy ever become available for all patients that need or want it? Can this specific special procedure ever be done by a hospital multiple times? Who pays for it then? There are also various ethical and social issues associated with gene therapy. People may have their own thoughts about gene therapy based on their own views on disease and health. It is also of importance to make a distinction in the public debate between gene therapy applied in the cells of an already existing person with a disease and gene therapy used to change the cells of an embryo that will subsequently be passed on to the embryo's offspring. The latter option is prohibited for ethical reasons all over the world because it is as yet unclear what risks are involved for the offspring. We all have to deal with the issues new and emerging therapeutic options bring to society. Since most healthcare systems are built around insurance for people with ill health, society comes to bear the cost of particularly expensive drugs or procedures. Only a couple of individuals could otherwise afford such treatments. As a society, we also have to deal with the uncertainties about the effect for the individual and how long that will last. It will take a lot of time before we know for sure what the exact effects on the lives of patients have been. It does not seem sustainable now that gene therapy will be developed for the over 8,000 rare diseases that we currently know of. It is difficult to predict what the effects will be on the group of inborn errors of metabolism that currently have no therapeutic options and for which gene therapy seems feasible. To think about and discuss these questions, especially when there is no therapy available now, is important. Patients and parents are a part of society. So, to wrap this up, Without naming specific diseases, we tried to explain that for some metabolic diseases, gene therapy is or is becoming available as a treatment to prevent the symptoms of the disease. Of course, we hope that patients will benefit from these developments. We have tried to explain step by step how the addition of a substitute genetic code can result in a correct protein or enzyme being built in the cells of the patient's body. There are two forms of gene therapy, in vivo and ex vivo gene therapy. There are also two ways of manufacturing gene therapy, either as a medicinal product or a hospital procedure. Lastly, we touched on some ethical and societal issues associated with the development of gene therapy. We do hope this has been of interest to you. Thank you for watching and listening. We'd like to add more disease-specific examples of gene therapy as soon as they become available.